Hi, everyone. Thanks for coming. I'm super excited that we are going to have Dr. Hannah Joy Warren presenting to us today. So, Hannah is an NSF Office of Polar Programs postdoc at University of Washington in the Applied Physics Lab. And she did her PhD at Stanford, working on a lot of really interesting stuff about phytoplankton physiology in the Southern Ocean. But now she's been kind of looking more recently at the connection between phytoplankton and air-sea CO2 exchange. So I'm super excited to hear her presentation. And yeah, I think she mentioned that, you're happy with people sort of interrupting and having it be kind of a conversation along the way as well. So yeah, take it away, Hannah. Thanks for presenting.
Thank you so much, Channing. Yeah, I would appreciate any interruptions if anything's unclear or you have questions. Let's just talk about it and yeah, make it more of a discussion. This is all super new work, so I really would appreciate any feedback, especially if you're like, I don't believe that or I don't trust that. Let me know that now. This is work in collaboration with some great collaborators. Ayush Nag is an undergraduate that I worked with for 2 years, who's now just starting a master's at the University of Washington in computer science. And then Danny has been a wonderful data science or collaborator on the data science side. And Allison, Andrea, and Allie are all advisors of mine in one capacity or another. And I'm first going to introduce you to phytoplankton community composition. For those who know me, it is my thing. I love it. I think it's so fascinating, and I think it's vitally important in the Southern Ocean. So I will hopefully convince you of the same. And then I'll do a brief carbon uptake introduction as well, although I think probably everybody or almost everybody here is quite familiar with that. So forgive me if it's a repetition, just enjoy. And if it is new, I hope that I also convince you that it is a super fun and exciting thing to work on. So here's our cross section of the Southern Ocean. We have our mode waters, intermediate, deep, and bottom waters all flowing through in this highly stylized diagram. When we're thinking about air-sea CO2 flux in the more northern latitudes, so to the right here, we have our mode waters that come down, move southwards, and as they do so, they cool, and we can get more carbon uptake there, and then they downwell and take their carbon, the carbon that it got in it with it as it transits back northwards. A little bit further south, we have deep waters upwelling here. And this is old water that has lots of carbon. So this is natural CO2 outgassing. And at the same time, we can get some carbon going in that then gets subducted into intermediate waters there. And all of this is controlled by physical processes that we are generally, as scientists, as the scientific body, fairly familiar with. Wind influences this a lot, how much carbon can go in and out of the surface there. Ice cover can create kind of a seal and reduce some of that transfer, although we're learning recently not all of it. And then other things like mixing and salinity and temperature of the water have a big impact on air CO2 flux. So this part scientifically we have a fairly good handle on. And this is my part here, the biology that's growing at the surface of the ocean. And our phytoplankton specifically, we have different taxa here, which I think is really important and I'll get into. But this can help influence air CO2 flux by the phytoplankton taking up carbon from the surface water, and that we can have more dissolving into the surface. So now I'm shifting this slightly, but I will put back everything we've just talked about. Air CO2 flux influenced by physical processes and biological processes. And this is now the diagram where I'm going to kind of talk through the pieces of this work. So the big question I'm trying to answer here is, what is the relationship between phytoplankton community composition and air CO2 flux? So in many of our models, not every single one, but in many of them, phytoplankton is just biology that photosynthesizes, but they're not really all created equal. And that's what I really want to dive into and try to open that black box of just saying biology. In a lot of reanalysis or float work, it is the residual carbon is assigned to biology. So I think it's been treated as kind of a black box, and I want to open that up. So the first step of this is to characterize Southern Ocean phytoplankton community composition. And here we're going to dive into that piece soon. And then the second piece is to estimate our Southern Ocean air CO2 fluxes, look at that carbon, and then try to tie those two together to look at the relationship. So diving into my favorite part, the community composition. Community composition in the Southern Ocean has a huge impact on ecosystem diversity and also carbon export. And our two kind of beloved taxa here are diatoms, which are these gorgeous ones. They have their silica shells. They are eaten by krill and copepods, and those feed the upper trophic levels that we know and love, all of our birds, seals, whales, all of the big, beautiful stuff. Physis, on the other hand, here is gelatinous. It seems kind of like a jellyfish type structure. It is a carbon matrix that holds all of those cells in there, which are the little dark colored dots that you see. This is a colony. And Phaeocystus is only really grazed by salps, which are also super, super cool gelatinous zooplankton that can contribute pretty substantially to carbon export because salp package it as salp filter feed. They collect the Phaeocystus and then package fecal pellets that they often re-ingest. So they make these really dense and super carbon rich fecal pellets because Phaeocystus has so much carbon in it because that gelatinous matrix that it makes is made out of carbon. So we can see this more graphically. This is a paper from my PhD advisor, Kevin Arrigo, from 1999 in the Ross Sea. And this figure in this paper is what really stemmed all of my curiosity in community composition. So this one is really important to me. Here we have nitrate on the y-axis and phosphate on the x. And as you can see here, Phaeocystus uses twice as much nitrate per mole of phosphate. So that line is steeper. This is also true of total DIC. I just changed that axis there. Unfortunately, Kevin did not make a plot of this, so I'm just using the nitrate one. But Phaeocystus uses twice as much carbon per mole of phosphate as do diatoms, and in making those gelatinous colonies. So this is really what got me to thinking we can potentially see a difference in air-sea CO2 fluxes if the phytoplankton there are using different amounts of carbon in the surface ocean. So that is all of my inspiration for why I work on this community composition question. Now we're going to dive into the science of it. This is a paper called Phytobase, a global synthesis of open ocean phytoplankton occurrences by Righetti et al. From 2020, which incredibly compiled data from three main sources: the Global Biodiversity Information Facility, GBIF, which is in the darker blues. Can you see my mouse? Over here. The Ocean Biogeographic Information System, OBIS, in red. See a lot of those points around here. And then MAIRDAT in this light blue here. There are some yellows, and that just means that they were in multiple sources. And so to look at community composition, I'm taking this Phytobase data set that was already compiled and looking at the Southern Ocean south of 30, trying to put the biggest bound on it I possibly can, just to look at everything that we have. And I'm gonna note here that I did not, this is Phaeocystus that I popped up, I did not use this, the taxonomy that they gave exactly, because the taxonomy of Phaeocystus changed, I think in 2017-ish, and it used to be separate from coccolithophores, which are our calcifying things. We think of the Great Calcite Belt. And now they are in one family, the Coccolithophyceae. So my first iteration of this, I used the taxonomy as it was, but things weren't quite right because taxonomy has shifted. So I updated the taxonomy of Phytobase. So I did not use it straight as is. And now here on the left, this is just a Southern Ocean map showing all of those points from Phytobase in black. And another note is that I'm in progress working on making my own phytoplankton database that I'm calling SOFI, Southern Ocean Phytoplankton Database. And Phytobase only has microscopy observations or cellular things. And I'm working on adding an HPLC chemtax, which will be the primary data set because we have a ton of that, as well as imaging systems from things like FlowCam, IFCB. And this is what my student has been working on for 2 years and is now just starting his master's, which is really exciting. So that is upcoming also. And if you have any Southern Ocean phytoplankton data, I would love to hear from you. I am trying to collect from all the cruises, make something that will be useful for our whole community. And because as you saw in Phytobase, it's only those bigger databases that they put together. So trying to collect all the little things that are missing. So one of the big puzzles here in connecting things is that BGC floats are where we have our carbon data, which is wonderful. They are not coincident in space or in time with our phytoplankton observations. So the black dots on the map on the left don't match up with the gray dots of our float locations. So this is kind of the first puzzle to solve, and how we're going to connect this data and use it. And to do this, I'm going to model phytoplankton distributions. And to do that, I'm using something called max-ent species distribution modeling, which I'll get into. The first input for this is the Biogeochemical Southern Ocean State Estimate, BSOSI data. And this, I'm using iteration 154, which is 2013 to 2023 monthly. So this is one of the big updates I just made. And we're looking at the mean in the mixed layer. So here is data from BSOSI in the spring and the summer. I've defined spring as it's kind of spring, late spring into very early summer. So it's October, November, December. Summer is kind of like peak summer through very early fall, January, February, March, April. I made those divisions based on the data that we had in Phytobase. So we had a peak in those spring months and then a dip and then a peak in the summer months. So it kind of, the Phytobase data naturally divided into those two. And I'm sorry, I forgot to put a plot of those. But the inputs for this model from BSOSI, which is based on largely on flow data, so that's why I chose BSOSI, sea ice concentration, temperature, salinity, oxygen, nitrate, and chlorophyll. And it's kind of nice, we can see in some of these pretty big differences between the parameters from spring into summer. Some of them look fairly similar, which is also okay. And I'm pairing this with our Southern Ocean phytoplankton observation. So this is the Phytobase data set with my changes to it. And so we have our taxa observations, and then we have what the properties looked like during those observations. And we can put this into the max-ent species distribution model, which is, here are inputs kind of graphically shown on the left, and then we get our taxa coming out to the right there. It's a probability distribution model. So how likely are we to see this taxa, given that when it was observed, the situation looked like this? And so here are the outputs. We have diatoms here on the top with the probability distribution, or the likelihood of presence from 0 to 1. 0 in the dark blues at the bottom, and then highly probable of finding it in those yellows. So sort of south of the polar front, we see a lot of diatoms, which we would expect. North of the polar front, the silica concentrations drop off dramatically, and we tend to not get silicifying phytoplankton like diatoms. Phaeocystis here is pretty abundant. There are some belts of it in the sort of subtropics. And then we see a hotspot just outside of the Ross Sea. And then we see within the ACC, we also get some higher Phaeocystis, which I think makes sense. Phaeocystis tends to be pretty good at using dynamic light. So in deeper mixed layers, when things are a bit more wild, Phaeocystis can thrive. Diatoms really like their shallow mixed layer depth. And so we did this for the 5 most common taxa in Phytobase. And again, I'm showing spring across and then summer. And then we have our taxa in the columns here. Diatoms, we were just looking at diatoms in the summer on that last slide. In the spring, we have less likelihood of finding diatoms, which again makes sense. We still had some ice cover in that time period. We haven't quite gotten things lit at the surface and going. In the spring, Phaeocystis is found more in those outer rings where we have more light, less sea ice. And then in the summer, it gets kind of more distributed throughout. Coccolithophores don't look quite perfect. We might expect to see our great calcite belt around the subtropics there north of the polar front. And maybe we see that in the spring where we have like fairly similar abundance of coccolithophores, not so much in the summer. And here I will point out, I mentioned that Phaeocystis was now within the family Coccolithophyceae. And so here I've pulled them apart just so we can look at it. And we have Coccolithophyceae, the family, without Phaeocystales, the order, which is the order that Phaeocystis is found in. And all of the Phaeocystales in Phytobase, all of the Phaeocystis was Phaeocystis antarctica south of 30. So we can really just think of that group as Phaeocystis. Dinoflagellates, not super abundant. I think that's okay. We have them, and similar with cyanophytes, which are kind of small little green things that tend to pop up when other things are not around, which we do kind of see they seem to occupy some of East Antarctica where we don't see a lot of other likely abundance. So here's our modeled taxa, which we can now match up when we get to it with our float data. So that's why we did the modeling here.
Hannah, can I just ask one quick
Yeah.
question? First off, like, because I don't really know about what the max and species distribution model does, I don't have a sense of like, do you know which of those input parameters like has the largest control or and that's kind of my first question. Then the second question, this is just like presence or absence probability. This has like no information about like biomass or anything, right?
Correct. No
Okay.
biomass. It's just likelihood of finding it. So like I kind of think of it as how good is that niche for that taxa.
Hmm.
So yes, this doesn't really tell us how much there would be, just that like it could be there. And then your first question is we do get output about the input parameters, which might have been fun to look at and could be something to dive into. The and the input parameters were here on the top left. Chlorophyll was the least important, which I actually think is kind of good because it's also the least confident we have data we have in BSOSI. So not terrible that it was the least influential of the distribution. Salinity was also very, very low. Sea ice concentration was quite important, and temperature and nitrate and oxygen kind of fell in between, depending on the taxa and the season. They all changed. We also get outputs of standard deviation. These are means, I should have mentioned this also, these are means of 10 runs, which is good because there's some randomness in there. And the standard deviations all looked fairly low, which on the one hand gives me some confidence. On the other hand, is the model overfit? I am not sure. And I think a lot of my confidence in this comes in having a fairly good sense of what distributions we see naturally and where the observations are. But this is all just modeled, so it's all kind of inherently not correct. And for now, I think it's the best we can do to try to start looking at this question. In situ studies are also great and kind of give us much more detailed small scale pictures, whereas this view is giving us like a broad brush strokes over the whole basin. What do we think might be happening and where should we maybe dive in a little bit more?
Great, thanks. I think Theo also has his hand
Yeah.
raised.
Yeah. Can you hear me okay?
Yeah.
I'm on my balcony, so there might be a bit of background. Um, Firstly, it's really amazing already. But I was just thinking in terms of the sea ice, if you're using the SOSI output or the BSOSI output, I know that for SOSI there's quite a lot of discrepancy between the observations when it comes to time series. I'm not sure how that. I assume it's the same when you come to climatologies. If that has a large influence, do you know if it's better to use observations to feed your model, or I guess maybe for consistency is SOSI better? Have you thought about this?
Yeah, it's BSOSI is far from perfect. I think that it's the best we can do because the observations are like our phytoplankton taxa observations do not have associated anything data. If you're compiling them from a whole bunch of sources, it's really hard to have any consistent data. And the associated data gets better in like the 2010s, but is pretty spotty before 2010. And so it's kind of a game between do we want more associated data with our observations, or do we want more observations? And we already have so few observations. So I've tended to the let's look at everything. So yeah, I think in situ data would be way, way better. We just unfortunately don't have it in the same space and time as we have our phytoplankton observations. But I do hope that, you know, towards the future, we as a scientific community get better at collecting things simultaneously and then archiving that data so that it is findable and accessible. Yeah,
Maybe
so I agree with you.
maybe in sync.
Yes, in sync, Antarctica in sync.
Mm.
But if we could just have way more of that all the time, and we had always been doing that. Yeah, I
Yeah.
have a lot of hope for the data of the future.
Yeah. Cool. Thanks, Hannah.
Yeah, thank you for that question. I did go back and forth on this a lot. Yeah, and it's not perfect, and not even all of my advisors agree that this is maybe the best way to do it. But it's what we have, is sort of my attitude. Okay, so another way we can look at these maps is by overlaying the probability of finding something. And so here, this is all 5 taxa on the top left, and then it's colored by the number of taxa above 50% likelihood. So you can see in some of those like kind of gray purpley, the really dark color, here we have like nothing is dominating there. Which is fine. Sometimes we have one taxa. This looks like it's probably the Phaeocystus, and this top one corresponds to this top column. So this is spring here. And then towards the continent within the sea ice zone, we get multiple things that could have a high chance of being there. We can do this for the summer also, where we see much more spread in where we have one taxa, and we have more of those highlighting where we might even have 5 taxa probable of finding. We can also look at just diatoms and Phaeocystus, because I kind of motivated all of this where we're interested in diatoms versus Phaeocystus. And so in the spring, we have almost nowhere where both of them are, which would come in a blue color, and then a couple places where one of them are. In the summer here, some of those blues are coming up, and much of the region has at least one of them, and not always the same one. So if we look like in here in the Waddell area, there's one taxa around there, which probably diatoms, but then up in here, we can also get a likelihood of Phaeocystus. You can see there close to the sea ice edge, same in the Ross Sea. So we have a much greater chance of being able to compare what diatoms and Phaeocystus are doing in the same region. And as I noted earlier, like we don't always circumpolarly going in and out, we don't always have the same carbon fluxes happening. So we don't really want to compare the subtropical zone to the sea ice zone, for example, if we're thinking about how is it impacting air sea CO2 fluxes. So we're going to focus in for the rest of the talk on the summer with diatoms and Phaeocystus, which is this lower right plot here where we can really get towards comparing the two. So a lot to really get at our phytoplankton community composition, but I think we did it. And next we are gonna try to think about our air sea CO2 fluxes, and then last stage is to connect those two. And so when thinking about air sea CO2 fluxes, how the phytoplankton are actually impacting that is really through the surface ocean DIC. So here I've plotted from our float data mean DIC in the mixed layer. Closer to the continent here, the sea ice zone is marked in this lighter blue color, and then we have our friends going outwards. Spent a lot of figures, and I should have said that a while ago. But we have you know, some lower DIC out here in the subtropical zone, some higher around the ACC. So similar to what we would have expected from the very stylized diagram I showed at the beginning. So now we're going to be comparing our taxa to our surface mixed layer DIC to get at how phytoplankton might impact the air sea CO2 flux, but I'm not going to actually show air sea CO2 flux, so sorry for that. And then I also popped this phytoplankton location map back up because you will have seen there are pie sections here in the different colors. and I've colored them by the fronts as well. And it's been fairly typical to divide the Southern Ocean when we're thinking about regions by climatological fronts. So those are shown in the white lines, and then sectors, which are just lines of longitude. And these flux a little bit based on the paper and the study, but this is a pretty common way to chop things up. And as a biological oceanographer, my first thought is climatological fronts, like, but who knows where it actually was when we were sampling that. And then as I've delved into the physical oceanography world, learned that fronts are not lines. They're not like hard boundaries that are really hard to cross necessarily. They're just, they're connected throughout, but they're not just one thing. So I started thinking, I really don't know that this is the best way if we're going to try to think about how the biology and chemistry and physics are all interacting to draw these hard boundaries like this. So a big piece of this project has been implementing a machine learning method called Gaussian mixture modeling. That is an unsupervised learning approach for clustering profiles with similar temperature, salinity, nitrate, oxygen, and density. I picked those variables. You could pick other variables that you thought were important for your question. So this should hopefully group profiles into biophysically similar areas. So then we can compare within those biophysically similar areas. And if we then see different DIC within those, we might be able to, say, ascribe that to a taxa. It's big foreshadowing right there. So here is just one float profile that I've shown in this map on the right here. This is just temperature plotted over time. This float went from 2017 to summer 2020. And we have our lovely profiles through depth, space, and time. And so we can take all of these profiles and match up who looks similar to each other and then call that a cluster. And here it is. This is the result, which I thought looked amazing. And I put our climatological fronts on there so that we can compare the region to the front. And I'll walk through each of these regions one at a time because they're a little hard to look at all together. So first, close to the continent, I'll work out, we have the sea ice zone, and this is, it might be hard to tell, the blue dots for the sea ice zone are very coincident with our climatological sea ice zone. This is a satellite sea ice zone, and it's not perfect. You can see kind of on this bigger map, plenty of these go out towards the next front, even slightly past the next front. But given that it looks very similar but not perfect is what gave me a lot of confidence in these regions. Next, and I've labeled these kind of in our canonical wording for these. We have the Antarctic Southern Zone, which I'm showing in green, that goes actually a little bit further outside of this front here. A little gap in data there, probably an interesting physical thing. Then we have our polar frontal zone, and this again moves a little bit further north, especially in the lee of South America, but very coherent still. Our Indian subantarctic, and I labeled this because it was largely in the Indian sector, but not perfect. We also have quite a lot of data there popping out in this region south of Australia. Our Pacific subantarctic, again, not a perfect name for it, but it's largely in the Pacific. And then our subtropical zone, which does cluster mainly into the Atlantic sector, but we have points circumpolar there. And it's really the main points that pop up in our subtropical zone. So this is now how I will group the data to look at things and start comparing them, which I thought this was super cool. This is the piece that I collaborated with Danny Jones on. So moving these to the bottom, and then I'll show data for each of these regions. So this is the mean DIC in the mixed layer. The light blue here is the sea ice zone, and these are slightly funny plots. They're called box and plots, but I really like them. They're basically a box and whiskers plot, but with those tails detailed. So instead of just a line where you'd have your whisker for your box and whisker plot, these are shown in quartiles going out, and we have our median in this thicker, darker line here. So DIC in the mixed layer of the sea ice zone. We can do this for all of our zones, and I can look at how different they are. Here, the curly equal sign means they are not statistically significantly different. And with the star, they are statistically significantly different from each other. And then everybody not next to each other, all of those zones are all different, except for these three were not statistically significantly different, but every other not next door one is different. And so this is really the motivation for why we need to look within one region to start comparing our taxa, because if we compared here CI zone even to Antarctic Southern Zone, I wouldn't know if the difference in DIC is because of taxa or because it's a different place with a different DIC. So now we have really characterized our DIC here to imagine how the air CO2 fluxes might be different. And the final and most fun step is connecting these to each other. And here's where I think it gets really, really cool. So here is the Boxen plot again of the polar frontal zone, just as an example, but we can divide that into where we have diatoms dominating or phaeocystis dominating, which I am describing as sort of like, are we likely to have diatoms here? Are we likely to have phaeocystis here? And that parameter I came up with was it has to be greater than 0.5 and more than 0.2 greater than the other one. So some of our float locations will have neither of them dominating. You could easily imagine this out here in the subtropical zone, in the northern section here of the subtropical zone. And you can't have more than one assigned because it has to be greater than 0.5 and greater than 0.2. Oh, sorry. No, you could in theory. I don't think it happens, but in theory you could. So you have to really be much more. Hold on. No, you couldn't if you have to be more than 0.2 and 0.5. Anyway, I'm sorry. Turn myself in circles there. And I'm coloring diatoms in this teal color. And again, forgive the colors because I did this very recently. Phaeocystis is in this kind of bright pinkety, like raspberry color is what I've been thinking of it as. Polar frontal zone is in this like purpley, slightly pink color. So we won't see them too much together, but I apologize for the color choices here. But we can divide our DIC in each region into whether we are likely to find diatoms or whether we are likely to find phaeocystis. This is now the same exact data, but as a split violin plot. On the left, we have diatoms. On the right, we have phaeocystis. And so I'm gonna now show all of the same data as the Boxen plots, but as the split violin plots in our same regions. And you'll notice in our three polarmost regions, CI zone, Antarctic Southern zone, and polar frontal zone, we have places where we could have either taxa more likely to be. And you can see that closer to the continent, south of the polar front here, we could have diatoms or we could have phaeocystis, or we could maybe have both. In the more northern regions, subantarctic zone, Indian sector, subantarctic zone, Pacific sector, and subtropical zone, we're only seeing profiles where phaeocystis could be, is more likely to be. So, and as we can see in this map, we really don't have a chance of finding diatoms out there, which again, we knew from the literature, we don't have enough silica there to get diatoms. So this is all kind of checking out with what we would expect. Now, to dive into the data, I think this is so cool. And here's where I got really, really excited. We have our DIC on the y-axis here, and then our sectors. And in both the CI zone and the Antarctic Southern zone, we have a significantly lower DIC where we have a likelihood of phaeocystis dominating. So this is all statistical relationships. This is not for sure what's going on. But if the statistics are telling us what we might expect, or you know, what we could expect to see there, I'm thinking here, phaeocystis is using more of that carbon in the surface water to make its gelatinous colonies than diatoms, which are using standard amount of carbon. And so we'll have lower DIC in the surface ocean when phaeocystis is there. And that means we have a chance of more air CO2 flux into the surface ocean, pending everything else like temperature, wind, atmospheric, PCO2. But the piece that the phytoplankton can potentially affect, we see a statistically likely effect here. So this was just, I was thrilled to see this. This was really really cool. And I mentioned to some people at the beginning that I've just redone all of this with more updated data, new iteration of BSASI, new, more recent float data. And we also saw this in the previous versions of the data, but with kind of less dramatic differences. But it also demonstrated to me that we're really seeing something that is likely to be true here, seeing it, doing it with multiple different iterations of data. So for the rest of the talk, I'm going to zoom into the sea ice zone where we have our statistically significant difference here.
Sorry, Hannah, do you
yeah.
mind if I jump in before you
No,
move?
please.
I might have missed it. How did you do this calculation or approximation? Yeah, I was just wondering if it was based off the probability.
Yes.
So yeah, could you just explain the way you've gone from that to the
So I
DIC?
started with the Boxen plots here. More with these Boxen plots where I just plotted DIC in each of these regions, the mean DIC and the mixed layer. They're different between regions, and so the next step there was we have to compare within a region. We can't do this across the whole basin. Can't compare between regions because we have different DIC for physical reasons. So we're not really going to get at the biology question. Yeah. So then instead, I can divide the data in the Boxen plot, our mean DIC and the mixed layer, into what corresponds to diatoms and what corresponds to Phaeocystus. And this I did using these probability plots here. So each, like the latitude and longitude of a float profile got assigned whatever the probability was there from the max-ent model. And then, but it only got called diatoms or Phaeocystus if it was at least 0.5 probability of presence and more than 0.2 greater than the other one. So it had to really be there and the other one not there. And so
amazing.
that
Thank you.
Yeah, no problem. I'm glad you asked for clarification on that. So that's how we divided the DIC within each region into Phaeocystus or diatoms. And again, this I did because I didn't want to compare between regions because there are so many physical things going on there that would be much more likely to be making the difference than our biology. And so then to like really interrogate this, to say, what's going on? Let's look a little bit more into this. Do we really believe this? I dove into the sea ice zone here. And so this is the same data. I tried to line up the axes, not perfect, but I tried. So our teal diatoms all of the data in that split violin is mapped onto the left side of the scatter plot, and then the pink Phaeocystus is on our right side. And on the x-axis, we have the probability of presence. So 1 is very, very likely to find a diatom there. 0 is like, there will be no diatoms here. And then the y-axis again is DIC in the mixed layer. And I've thrown on just some linear regressions here and plotted or written on there the intercept. If we had, for example, 0 diatoms, we might expect we'd have a 2120 millimoles of carbon per meter cubed of DIC. And then our slope in DIC for DIC was 86. So that means that if you went from 0 probability of diatoms to 100% likelihood of finding diatoms, we would expect 86 millimoles of carbon per meter cubed difference in that water. So I just mapped that up and rounded the number slightly, which is why the math is not perfect. Same with the Phaeocystus. So
Hannah,
if we have, yep.
Sorry, Carla here. I have a little question. I'm not sure if I'm interpreting this okay. So the DI, it's kind of like the probability that the community that is taking up this DIC is diatoms, right? That's the train of thought.
Yeah. It's really, that might be one step too far because this is all just statistical analysis. So I can't really say who's doing it. But what we can say is in water where we would have DIC around 2100, then we also in that same region expect there to be no diatoms present.
Okay, perfect. Thank you.
And one great argument here is that how do we know that it's not an entirely different factor that's affecting both the DIC and the taxa, and it's not the phytoplankton affecting the DIC. And I don't think that we can answer that with statistics. I think we need to go and do in situ observations like Theo suggested, and like I hope we will continue to do in these regions. So, you know, there's,
Thank you.
yeah, there's no causality that we can say here. I think there probably is causality, but that's just me, not the data.
Yeah. Figuring it out step by step. Thank you.
Yeah.
Is this
No
the
problem.
year-round data though, or is this only for spring and summer? Because yeah, you might expect like even if there's a high presence of diatoms in those sort of static maps, if the profile itself was taken in winter when there is, you know, really low production, then that's one way where it might be, you know, correlation and not causation. Whereas if you were only looking at data from, you know, the spring and summer when we might expect there to be active, you know, production happening, then maybe the causality could be argued more
That's a
directly.
good point. This is year-round data. I did the exact same
Hmm.
plots with only summer data, and it looked the same. And
Cool.
to my, yeah, maybe I should only do growing season like October through April or something. I was thinking that like if we had diatoms there, it you know, we would still see that signature
Yeah.
months later versus if we had Phaeocystis there. So I think it can be argued both ways which data to use, and I have these plots for the other one. I should have put them in backup slides. But the story does not change.
Yeah, I mean, I totally agree though that you still could make the argument that even if there's no active production happening at that time of year, that there still could be an effect on the mean that has to do with what happens, you know, during the growing season. Yeah, I think that's certainly like possible.
Yeah, so I agree. I've gone both back and forth both ways and looked at it both ways. The good news is the story doesn't change. The plots look the same. And the numbers change very slightly, but it's still significant. So fortunately, at the end of the day, it doesn't matter too much which way we look at it, which gives me confidence also. And then I have sort of one last summary of this data, one other way of slicing it up that again gave me a little bit more confidence. So you may notice, take a look at the DIC on the y-axis kind of spread across, and you may notice, this is something I picked out with my advisors. We were staring at it and went, hold on, there are kind of bins here. So there's sort of a high DIC that's like breaks pretty nicely at about 2150 millimoles of carbon per meter cubed. We have another kind of mid-range bin here that I call medium DIC from about 2050 to 2150 millimoles of carbon per meter cubed. And then we have kind of a third low DIC bin here, less than 2050. So I'm going to flip on its side here into these bins now. And so the bins are on the right, on the left is showing the data that went into that, colored by low, medium, or high. Again, apologies for this color. We're back to purple versus pink, which is a little confusing. But all of it stands without colors too. So on the left side of this box plot, so now we have our probability on our y-axis, and then we have our DIC bins along the x. Flipped things on its head a little bit, but super cool, in our low DIC bin, so now we're kind of collecting a little bit more data together, we see a statistically significant difference between if we have a probability of diatoms present versus a probability of Phaeocystis present, where if we have high, in a low DIC bin, we would expect higher Phaeocystis presence, or more likelihood of presence of Phaeocystis. In our medium DIC bin, we would expect higher probability of diatoms, lower probability of Phaeocystis. And in our high DIC bin, that's even more exaggerated, although this one was not significant. But we kind of see this stepping up in probability of diatoms as we go to higher DIC, and stepping down of phaeocystis probability as we go to higher DIC, which you may recall is the same thing that we saw here in our linear regressions. Just found it to be slightly more clear in these bins here, which I think is just so cool that we can see that when we have low DIC, we have a high probability of phaeocystis. And again, this is a statistical relationship. This is not causal, but if I'm thinking scientifically about how this might be happening, we're within the same region. So and the regions were defined physically. So we don't probably have a ton of physics influencing this, probably some, but it may not be the dominant factor. We do have higher chance of finding phaeocystis, and we know that phaeocystis uses more carbon than diatoms do. Of course, a lot of this will depend on the actual chlorophyll. So like not just who might be there, but how much are they photosynthesizing? Because we can have a lot of diatoms and low phao, and then diatoms will be using more carbon. But I'm hoping that grouping such a huge amount of data together will help us kind of, all of those other things will even out. So this is not finished, as you can see. This is kind of the state of where I'm at. And with recent updates to newer data sets, we saw this in the older iteration of BSOSI and an older snapshot of float data. But I'm really excited about this, and I think that this really tells us that there could be something here, and mostly we need to look more into this. We need Antarctica in sync. We need to get out there in the field and measure these things. So this is just back to my diagram as a small summary here, where we've connected Southern Ocean phytoplankton community composition to our mixed layer DIC as an indication of how phytoplankton might be influencing air sea CO2 flux. And we've actually seen a relationship there, which when I started, I thought could be possible, could be a thing that was happening. I didn't know if it was going to be possible. And it took combining a bunch of very disparate data sets using machine learning and modeling to bring these to like get data that was at least semi-comparable to do this. So huge number of caveats especially in modeling and machine learning, and still we see a relationship there, which I think tells us we need to go look at this more in depth. And so with that, I will wrap up and take any questions.
Thank you so much, Hannah. That was so cool. It's really cool to see how this work has kind of been evolving over time too, since I've seen you present it at a few different stages. One thing, maybe I'll just kick it off actually with a question, is that, or it's sort of more a comment, even something to just like keep in mind to connect this to the air CO2 exchange, is that it's really striking to see how much of a relationship there is between the mixed layer DIC with this like diatom versus phaeocystis dominance, particularly in the sea ice zone and the Antarctic southern zone. And those are the two frontal zones where we might expect the DIC component of surface ocean PCO2 to be dominant because we're at high latitudes, because, further north, the thermal component could be more important even. So I think it's just even like a further indication that this could be really important actually to the air sea fluxes, not just to the DIC that you're showing here. But I think like the places where you're seeing those differences in DIC most strongly are actually the places where we might expect the DIC to have a really strong control on the surface ocean PCO2 and the air sea CO2 flux. So I think it's, yeah, just thought I would mention that because I feel like it's just sort of something to even strengthen further, like the connection to the air sea fluxes between what you're showing here. But yeah, it's really amazing work. So thank you so much.
Oh, thank you for that comment. It's
Mm,
very helpful to have the
mm,
physical
mm,
oceanographers and chemical
mm,
oceanographers help
mm,
direct my biology brain
mm,
to like
mm,
what's really going on and what's really important. And yeah, I appreciate there have been a lot of
mm,
iterations of this. You're right.
mm,
And I think as I go to the more updated data, it's just
mm,
getting stronger, which is
mm.
fun to see.
Cool. I think
Lily.
Lily, yeah.
Hi, Hannah. This is amazing as always. And I just have some curiosity questions, but I want to acknowledge that they are coming like, oh, what do we do next? And I want to acknowledge that like there's a lot of work that's gone into this and we should celebrate that. But thinking about like how this, one, do we have an understanding of how the like distributions or concentrations of physostis and diatoms have like changed in different like climate states, and can we do any projection into the future? And then the second question is like there's a lot of talk about like different like nutrient limitations across the Southern Ocean. I'm wondering if your new beautiful shiny like SOFI data bank can help us disentangle some of those questions.
Great question. So first, do we see changes with changing climate, and or what do we think about the future? I would say in situ data, not really, because we just don't have in situ data for, there is data in this data set going back to, I believe, 1860. However, not in the Southern Ocean, in phytobase. In the Southern Ocean, I think it starts in 1920s, and there was like, 5 points in the 1920s.
The water was a little green.
Yeah. Yeah. So we don't get, I think, super reliable data until we have some in the 60s. We have virtually no data in the 40s because of the World War, which I thought was fast. There's nothing in the 1910s and nothing in the 1940s. We have a little data in the 1920s, and then we have some going from there, but I don't think we're getting deeply reliable data until we're getting into the 80s, 90s. And even then, it's like you're getting a few points in the summer, and we're not getting any kind of seasonal cycle until the 2000s, really the 2010s. So I don't think we can see it in situ data. I do think that we can, that's where models are phenomenal and where we need the models. And we do have plenty of lab experiments that show in these conditions, this is how things change, which I, in a former life, was a phytoplankton lab person. We don't have any of the interactions we see in the environment, even when we do interactive experiments. So I think that we can project into the future based on lab work, which is, and a lot of my PhD was actually ship-based experiments, so that we are at least taking water from the ecosystem and looking at it in the temperature, in the solar radiation that it would have been getting. So a lot of my PhD was thinking, who are we going to have in the future and where? And we have big seasonal changes in which taxa is dominant, which is also why I started looking at spring and summer. You can look at January of every single year and totally miss taxa. So long answer, yes, we will be seeing changes. We do have data to look at this. I was doing ship-based experiments, so I would love for a modeler to use my numbers to parameterize the model and actually look at the future, instead of me just thinking about what that might imply for the future. But, and mixed layer depth, I mean, very canonically, diatoms, shallow mixed layer depth, physostis, deep mixed layer depth. Which way is the mixed layer depth going? Down, but up maybe, and regional, and we're not really sure.
Yeah.
So I think it's, there's pieces all over, and we just need more data. We need more field work. We need more modeling, which I think is the summary of every Southern Ocean project.
Yeah. Hey, Hannah, it's Carla again. Well, and in sync, we trust.
Yes. Yes.
I have another little question.
Yeah.
My work, it's mainly in the very unseen eukaryotes in winter time. So I'm very happy that you're creating already this SOFI database. I think it's very, very cool that we gather the Southern Ocean data together. And my question is like, if those cyanophytes that you highlighted as a fifth community would be mainly pico carrots?
That's a great question. I took the labels that were given to them in phytobase,
Okay, perfect.
were taken from obis, meridat, and OBIF. So I don't know.
Yeah,
I,
probably just a size cut then.
yeah, which I think is problematic. I think this is one of the biggest gaps maybe in models that do anything to say anything about taxa, is they use size categories. And Phaeocystis, one cell of Phaeocystis is 5 microns. Colonies you can see with your eyeball, like it can be a few millimeters. So I don't think that size is always deeply meaningful, especially in the Southern Ocean.
Mm. Yeah, thank you. Great work.
Question. Thank you.
Oh, this actually relates to a question that Tiago asked that I didn't see earlier, but about whether it's possible to separate diatom groups by size um, using this data. I'm not sure if you were talking about the MaxEnt model, Tiago, or just the database itself. But
So the MaxEnt model is like gives you whatever you give it. So you could give it size categories and then it would output size categories. So that's kind of whatever you want it to be. The database, phytobase, does not have anything besides we saw this thing here. So it's a presence only database, which is kind of as good as we can do with phytoplankton, because nobody's writing down, we didn't see anything else. We only saw these things. And they did not include anything about size, which is in my database. I am trying to compile all of that ancillary data that would help us answer these questions. But you can only put in what researchers report, and I think it's not terribly common to report phytoplankton sizes. With some of the imaging systems like IFCB, FlowCam, a few other things do this too, will give you sizes. But then you still have to choose to save that data and put it in your data set. So sizes are hard to come by in field data. I think in lab data, they're far more common.
yeah, I see he just responded in the chat and said that he was actually just talking about the distribution maps from the MaxEnt model. But great. Well, I think we're actually out of time already. So thank you so much, Hannah, for speaking. It's really amazing work. And yeah, I mean, it's really exciting results too, I think, especially, yeah, that difference between the Pheo dominated versus the diatom for the DAC value is like really striking and seems to make sense, at least with the biology. So yeah, it's really cool.
Thank you to everyone for coming and listening to me talk about phytoplankton. It's my thing. And yeah, I was kind of shocked to be able to actually pull this out, despite expecting that it could be what was happening. So thanks for
Great.
listening, everybody, and thank you for all the questions.
Okay, bye everyone.